Okay, welcome everybody. Happy Friday. Happy slushy day. Holy cow. All right. We've got just a handful of slides left to wrap up the tail end of chapter 12, thinking about some of the biotechnology and some of the techniques or tools that we might be able to use to evaluate DNA or the protein that the DNA, the genes within that DNA are coding for, which is right where we left off with the SDS page or polyacrylamide gels. There are lots of potentials depending upon what sorts of stuff we're really trying to evaluate, or we may be interested in the product that this organism, whatever it might be, could be producing. Running it out on a page gel gives us some details about some of that, some ideas about sizes of the proteins and really amounts that we're looking at, how many different types. And we talked about this in a little bit of detail. There are some other techniques where we may employ some immunology and do some Western blotting where we can take these proteins and examine them versus the Southern when we're thinking about looking at DNA, all right? And some of these proteins, and we'll hit some immunology in our next chunk of material, but some of these proteins we might think of as being antigens, and an antigen is something that can stimulate our immune response, and our body sees it and says, that sucker's foreign. I'm going to do something about it, right? I'll make some antibodies. And hopefully, if I see it again, we'll recognize and we'll mount responses, right? So knowing that, we could have some antibodies that have been generated against a specific pathogen, and then we could sample and see what proteins truly are responsible for the production of that in the first place. And that's really what some of the utility of Western blotting does for us, giving us some opportunities to evaluate these proteins. Likely a good chunk of these that we see on the gel, and this is just full of stuff, but some of these gels, we may see a handful of bands, and then when you do the Western blot, only one or two of them actually showing up, because it's those that are the antigens and stimulate the response. The other proteins don't, and that we may like to know a little bit about. Could potentially set the stage for vaccine development or other opportunities, right? There's some other techniques where we could evaluate this, and there aren't, there's a little bit of detail in the book, but not images in here, but we could think about running a gel to look at proteins, and instead of just this SDS gel, it's a kind of a two-step process with treating this, where we run it out and separate based off of size, and then we separate based off of charge. And in doing that, it's a native gel, not an SDS gel, that's going to denature and make everything the same, right? So we separate one way, and then we express it to current go in the opposite direction, and we can see things moving out. So you wouldn't run a bunch of samples simultaneously. You'd run one lane, and it would move down, and then it would move over, giving you some ideas about properties of those proteins. Some are neutral, some are basic, some are acidic, some are positive, some are negatively charged, all sorts of different potentials. And a 2D gel gives us some more insights, right? So as we transition, and there are handfuls of things in some of these chapters that I'm like, well, this is just flat out of place, all right? Sort of not, but in the PowerPoints, it definitely is. I'm like, now we're talking about Lyme disease? What's up, all right? I'm gonna back, I gotta stop this first. I wanna dive to the textbook real quick, because I mean, I saw the bullseye rash. I know we're talking Lyme disease with that stuff, but my question was, what sorts of context are we really talking about here? And it does fit with what we're talking about in this chapter, but you've got no setup. So I decided we ought to maybe do a little bit of this setup. So this case study, I think, is pretty interesting. And oftentimes when we think about, Lyme disease is something that, it depends truly on where you're at as far as potentials. We don't really think about it around here, right? But in some areas, it's definitely more common, but maybe not something that's always on the radar, right? And that bullseye rash that we saw on that previous slide, that is pretty characteristic of Lyme disease, but it's not something that always is exhibited or expressed. It's a really kind of a rarity, right? Maybe 25 or 30% of cases actually show the bullseye rash, right? So the bulk of them don't show that. And that opens up the potential for all sorts of misdiagnosis, right? Thinking we've got the culprit and ultimately not, all right? So there's a lot of detective work that oftentimes gets employed when we start thinking about dealing with patients, and especially dealing with some cases that really kind of seem odd, right? There are, and we're talking about some bacteria as we're moving through this, but there are lots of times when some weird oddball things may show up in a clinic, and clinicians now are thinking more kind of outside the box a little bit. You may not be able to directly pinpoint some of this. And the thought is maybe we should look elsewhere. What about some crazy fungus? Maybe we should think fungus, right? And that opens up all sorts of ideas. And, the cordyceps and all of that. I'm like, what? Yes, right? Or no. All right, enough of that. So we'll go through this and look at it just quickly and scan through here. But we start off with a patient, a 24-year-old female electrical engineer and running enthusiast. That's your first mistake, all right? So in turn, she moved from Arizona to New Hampshire, takes a new job, and wants to continue running, it seems as if. So she's running through the forests, hiking in the mountains, checking stuff out. She develops a low-grade fever, a headache, some muscle aches, feels a little bit fatigued, all right? So she takes some ibuprofen, thinks she's just coming down with something, and gets some rest. I think most of us would probably think that seems like a common way to deal with something like that, right? So we progress though, and I'm just going to jump through these and talk about them quickly. So she's got mild flu-like symptoms, and there's numbers of things that she starts thinking, but she's feeling ill enough that it's time warrants going to a clinic, right? So there's non-infectious autoimmune situations that could be a potential for why she's feeling poor. MS or lupus, ALS, all of you are like, holy cow, I hope it's none of that, right? All bad news, right? And mirroring a lot of the symptoms that she's describing. So definitely on the radar, all right? And we will look at some of this stuff a little bit later when we get into some of the immunology stuff, all right? So over the course of a few weeks, her symptoms start to worsen. She's got joint pain, heart palpitations, strange limpness in her facial muscles, so a little bit of paralysis, stiff neck, headaches, and this is what truly warrants going to the clinic, all right? So part of this stuff, maybe the headache and the stiff neck, this lends to some alarm as she first shows up in there, and the clinicians, their first thoughts would be meningitis, right? Likely bacterial meningitis is something that they're thinking likely what she's got going on, all right? So I should have just skipped ahead, but I gave you that before, right? So that's what they suspect, all right? But they rule that out. There are other opportunities besides bacterial. There could be some viral culprits with that as well, but they're ruling out meningitis initially. All right. So a lot of her issues seem to not quite fit with the idea of meningitis. All right. So some of her other symptoms, we're still going back to this autoimmune ALS or lupus, systemic lupus erythematosus or amyotrophic lateral sclerosis. So Lyme disease is also a potential, all right? So they're considering all of these, all right? She doesn't recognize or recall being bitten by a tick and never exhibited the bullseye rash, which was where we started really today's lecture with, all right? So they ordered an MRI scan, blood count for anemia and other tests, kidney function, trying to look for lupus, confirming or ruling out that, or Lyme disease. Her tests inconsistent with lupus and ALS, and the initial tests with Lyme disease, trying to look for antibodies in her body against it, were unequivocal. So we couldn't say yes or no, all right? Inconclusive doesn't mean one way or the other, all right? So we still can't say about it, all right? So they want more tests looking for this, all right? And that ultimately, there's lots of potential issues with this. You know, a few, 30 or so percent may exhibit this bullseye lesion that you can see, others don't. And differences in the way our bodies might respond to infectious agents are definitely varied as well. Some of us may produce a very competent and rapid type of immune response and be able to detect antibodies, and others it may be more slow to progress. And it depends on, you know, maybe the amount of these organisms and the levels of antigens that are stimulating immune responses. And if they're just not quite sufficient at this point, you may go undetected. All right. So similar idea, like, you know, a rapid strep test that may show up as a negative, but when they cultivate it, the bacteria actually is there and grows up. There's sometimes tests like this aren't giving you the full picture, right? So here's that, back to that bullseye lesion that you can see. You see that, you know, you've got some problems, right? So as we progress through here, all right, her symptoms progress, they become serious enough that she's impacting her daily activities. So they order some more laboratory tests, all right? They want to look at her blood, her cerebral spinal fluid, the synovial fluid from one of her swollen knees. So assuming maybe she was bit on the leg by this tick, right? But didn't recognize, but that might be a potential, right? So they request some PCR analysis of this. So we want to try to amplify what we're finding within here. Maybe the level of, you know, antibody wasn't sufficient, but if this organism truly is present, maybe there are some subtle signatures of its DNA that we might be able to detect from these specific fluids, right? So as we move through here, the tests from the spinal fluid and the synovial fluid both come back positive for the presence of the culprit, Borrelia burgdorferi. It's the bacterium that causes Lyme disease. So they prescribed full course of doxycycline. And with this, it really, there's all sorts of issues and details with Lyme disease, but dependent upon circumstances, maybe the bacteria has, you know, persisted within here for a long period of time. And at this point, treatment becomes a big issue. But in these circumstances, it was caught relatively quickly and the full course of antibiotics truly did its job. But in some circumstances, there's some post-treatment Lyme disease syndrome. It's after you've already gone through your courses of antibiotics and you manifest adverse reactions that potentially are lifelong, all right? Definitely big concerns, all right? But she appears to be not impacted by that, but it is something that truly can cause issues, right? So, kind of interesting, right? And we will move through, I'll shift out of this, but as we move through and look at this last little bit, utilizing techniques like PCR in a clinical situation may give us an opportunity to detect things that we may not be able to detect otherwise. And in turn, like some of the COVID with PCR lending to the ability to detect that more rapidly. Okay. I could maybe just scroll down here. So these are some different recombinant products that have been generated. This table is not in the PowerPoint with some of their potentials. We talked insulin, some of the first stuff, but there are all sorts of others that we think of as that possibly lend potentials for different disorders, right? Like cystic fibrosis, hemophilia, and others. So I guess maybe I'll just stay on this because it's got all the same images, all right? There are some potentials as we move into this next bit, RNA interference technology. So there's a handful of different things that we may be able to link up with and stop productions of things, right? So in this case, we've got our messenger RNA that's been transcribed, we've modified this stuff, and now because here we're looking at a nucleus, right? Antisense RNA is a complement to this messenger RNA, and if it happens to be present, it's basically going to stop the translation process. It binds to the single-stranded message and basically makes it so that the ribosomes aren't able to recognize, and we can't build proteins. That's what we're seeing right here. So these in turn serve as a mechanism to stop or to mediate the production of some of these proteins, right? So thinking, potential for maybe being able to treat some types of viral infections by going in and targeting the viral product that they would be making, the messenger RNA that says, build me some capsids, the coats of my virus, right? And if you can block that, you could help the production of that, driving a wedge in the synthesis or the cycle of that virus. This next bit is another, some different potentials with some RNA that can interact. So some of these mimicking natural processes. So small interfering or microRNAs, they in turn interact slightly differently, but similar sort of an idea to basically take that messenger RNA out of commission. So making it so that the ribosomes can't get in there and translation's not able to occur because those microRNAs have linked up and blocked access, or the small ones, every time I'm like, the small interferings, it's like that antisense. It's a complement to it that is a perfect match, and it causes a double strand block on the message, and that causes the messenger RNA ultimately to get degraded or destroyed. There are lots of things happening inside of our cells that might recognize some of these potentials. If we've got faulty messenger RNA that has been generated due to some sort of mutation, we potentially don't want that to be trying to build protein, and we might try to destroy it. So these mechanisms can be utilized, flipping it around, thinking similar idea, like to prevent some viral progression. That's the idea with it. And I did leave it on this because really, I believe in the PowerPoints. I think this is like the last bit. If I remember right, I will click back over to it just to make sure. But as I was scrolling through, looking at those case studies this morning, just going through a little of that, this last section with gene therapy, I don't think it's in the PowerPoint at all. And I'm not 100% sure why it's missing. So there are numerous ideas and potentials, knowing that we've got the ability to examine, to detect specific issues, and that sets the stage. Could we correct some of those problems? And with gene therapy, we think about an opportunity to make corrections, to cure specific diseases, genetic diseases, right? We will talk as we see some risks on this heading. There are definitely some potential problems with this, and we'll see one as we move through here that really kind of put the brakes on this whole idea of curing. And now when we think about some of this, we still utilize some of it, but it's not a thought of a cure. It's more of a thought of therapy, trying to help to treat disorders, but not cure, right? I feel as if we're really near the point of being able to go back to thinking about curing, right? But still, there's definitely some issues. And there are other ideas with like CRISPR-Cas systems that bacteria utilize as kind of like their own sort of immune response to chop up viral, bacteriophage comes in and tries to infect, they try to chew that stuff up. And in turn, it recognizes specific things and makes, like an enzyme can make cuts, and it also can make insertions. You could use these to do all sorts of manipulating and designer sorts of situations, which are happening right now. And that really sets the stage for thinking about maybe other ideas. So one thing, and we've got right here with SCID, we will mention in the next chunk of material a little bit about this, but SCID is a genetic disorder, something you are born with. It's called severe combined immunodeficiency. It's not the same as HIV or AIDS as it progresses to acquired immune deficiency, but it's sort of a similar idea, all right? In the case of SCID, you basically don't really have an immune response at all right? And it sets the stage truly for you to not be here. Individuals, children born with this didn't make it very long. They get sick and die because anything and everything would make them ill, all right? So there was a kid named David the Bubble Boy. There's technically a couple of movies about this. He was born in the early 1970s with this disorder. Doctors recognized really quickly what was going on, and they put him into a sterile environment. He lived in a bubble. He lived in this big chamber that was sterile, and everything coming into the chamber was sterile, so that he never could come in contact with anything that potentially could make him ill. Because if it did, it's not going to be able to really get better, right? So it basically had no actual physical contact with anybody, because if he did, it was trouble, all right? And I know later on we will hit a little bit of SCID, but there's a picture of him where NASA built him a spacesuit, so he's got his own self-contained bubble and he could get out of his growth chamber, his bubble, that would allow him to kind of interact a little bit, but still in his own contained unit. He couldn't interact and touch anybody again. All right. Bad news. All right. So the hopes were that science would find a way to help him to cure these problems. And he lived for a little over a decade, and there was really no hope in sight. And there were all sorts of things that occurred where he basically decided that he was done. And he crawled out of his bubble and hugged his parents and got sick and died. Sad. We're leaving on a sad note. We'll talk about some other stuff. So shortly after that, couple of years, the first child with SCID got cured with some gene therapy, right? Which seems pretty unfortunate for little David, all right? But there were also problems, all right? And we've got a box down here with something going wrong. And this wasn't SCID, but there have been issues with a handful of these. And I think SCID was another one, all right? So 1999, this boy, Jesse Glassner, 18-year-old, who received some gene therapy. He was part of a case study to see a clinical trial, I should say, to receive this gene therapy for a disorder he had called ornithine transcarbamylase that basically made it so he accumulated ammonia in his blood, couldn't process it and deal with it. He should be voiding that out in your urine, but he wasn't, all right? And he received treatment, and 4 days later, he died. He had a reaction to the virus that was bringing the treatment into him. And that, oops, back up here, is right where this figure is talking about, all right? Again, another one that's not in the PowerPoint that's posted. So we talked about different ways that we might use abilities to get something in, right? We talked with bacteria, handfuls of ways. We talked about trying to manipulate stuff with plants, where we could use argobacterium, that bacteria, to infect, or gene guns, or some other opportunities to try to get plants incorporated with something else. And when we think about, our own cells, virus oftentimes is a good vector, a good mechanism to get the specific into that cell, right? Cells have receptors for virus to potentially link up to. And so we see some specificity with that, right? So in this case, this adenovirus targets these respective cells, gets brought into it, and then deposits the gene that's been manipulated into this cell, right? And now it's able to go through the process of transcription and translation, and you get the proper product. We've made a cure, right? And there were a couple of potentials where we could see more than just this one, where there were some cures, right? And then ultimately lending to issues, right? So it sets the stage for lots of questions, right? Ethical dilemmas, right? When we think about medicine, we're trying to help people, right? And trying to cure them seems like a brilliant idea. But if in the process of curing some of these individuals, we kill others, that's a concern, right? And that's really why the brakes were put on us. We're like, we can't try to do this until we truly have a better handle on how these things are functioning. Are we getting the proper amount into the respective proper cell in the first place to actually get it to do its job? Or are they targeting other cells? And some of the circumstances, I think, with the SCID, where the other cells ended up being targeted, and it resulted in different types of cancers that were generated in these individuals because of the therapy, right? So numerous things that are potential problems with that. there are, and I don't know if we've got other details in there, that's the end of that. There are other ideas. Instead of curing, there are some like some issues with your immune cells where we could think about treatments trying to help with these issues, where we may be able to pull the specific cells out, infect with the right gene of interest, harvested cells from your own body get transplanted back into your body, right? But these cells have been altered, corrected, right? Your immune cells, like your red blood cells, have a really pretty short lifespan, and a lot of the white blood cells, 4 or 5, maybe 6 months longevity. So lots of these individuals require treatment a couple of times a year to ensure that these cells are constantly the new proper ones and not the ones that have some sort of mistaken problems with them. So, I think this is interesting, but it's also a bit disturbing when we think about some of those potentials for it to blow up. Let me flip it back to here's my PowerPoint, back to the PowerPoint real quick and just make sure I didn't leave anything off. I know there were some images that I didn't cover. We need to hit a little of that stuff when we think about some of these potentials. We've talked a little bit about southern blotting. So here, running out on an agarose gel, the DNA, and then transferring it to that membrane where we can get specific probes that link up to segments of that DNA, giving us some insight. It's because I was before it. Some PCR reaction. And I also noticed, I'm not going to click on it. I'm not getting off of this one now. But right near this figure, there's an interactive. I don't know if any of you have tried it. Anybody? If you're going through the OpenStax text, you can click on this and see PCR happening and manipulate some of this stuff and see what really is occurring over the course of a handful of hours. All right. So with this, we've talked about some of the ideas. This gives you kind of an overview. A handful of cycles, only 4 cycles that we're looking at here to get a number of copies, and we would do this thousands of times over the course of a few hours to get visible quantities in that tube. So in our first cycle, we've got a little test tube, a microcentrifuge tube. It's got our gene of interest. It's got primers that will link up to the DNA. It's got all of the nucleotides needed, and it's got the polymerase that's going to get in and allow this all to happen. All right. So the DNA gets denatured. It gets heated up at about 95 degrees Celsius. It gets cooled down. It heats up super quick and it cools super quick. So 95 degrees for a second and then down to 50 degrees. At 50 degrees Celsius, this is the annealing. So we denature, we unzip, then we anneal. That's where the primer links up to its strand of DNA. And we've got both forward and reverse, as we see here, 2 different colors, right? So the primers anneal, and then we go through our extension phase. This is a little bit warmer at about 72 degrees Celsius. It's when the DNA polymerase is able to do its job, and extension gives us the new copies, right? And then now we're at 2, and we're back to the start, denaturing, annealing, extending, over and over and over again. All right. Breaking the bonds, linking up primers, building new copies, and we keep progressing. All right. And right up here, we see the initial strand that we dealt with. And then these are all of the new ones that we've produced as we go through there. So there are some techniques that we can utilize to kind of evaluate what's happening in here. And we can use this to get an idea about what bases are being laid down, all right? What sorts of sequence did we really generate? So we can get all of the specific nucleotides figured out. So this is a normal situation with 3 phosphates, a deoxynucleotide triphosphate, all right? Adenines and thymines and guanines and cytosines. They all come in with 3 phosphates. We'll pop 2 off as we build that backbone. We talked a little bit about that before, all right? So this is the normal guy, and this one's different, all right? Dideoxy nucleotide triphosphate has an oxygen that's missing over here that is present in our normal one, all right? Making it a little bit different, all right? And as it's trying to form linkages from one to the next, it can't, all right? And so it causes, if it gets incorporated into one of these pieces, it causes it to stop being built, and there you go, right? And the idea would be that there would be multiple, through thousands of these replications, multiple points in here where you'd go with your very first one getting it to finally the very last one, and it's all random as far as how that happens. And you get different sizes of chunks of DNA that have been replicated, right? So that makes it stop, but we need a way to detect it, right? How can we color code this to make it work? So there's some fluorescent tags that they link up to the respective bases, and they glow when this happens, all right? So the DD is the dideoxy, so it's the one that's lacking that oxygen, and it's also got this fluorophore, this molecule that will fluoresce when it gets excited. So each of these PCR products, from a little teeny bit to a great big full sequence, goes through the sequencer, shooting a laser, exciting the fluorophore, and dependent upon where it's at, it basically marks it off. It's kind of counting the nucleotides, but it doesn't know what nucleotide it is until it glows, right? And number 1 would glow, and finally number wherever we're at, here's 130. So now we're down to 140, right? Each one of them at some point would wind up with the dideoxy incorporating instead of the deoxy, and it's got the tag on it based off of its respective color that's on there, right? And as it moves through there, it gives us these respective peaks showing where basically where we're at, and spitting it all out, you get the sequence data, right? That tells you here's this whole product, right? And when we are trying to understand like genetics, we may extract DNA from something. We may ship that off for sequencing, and this is basically what happens. They run PCR products, running it through this mechanism, linking it up to different tags, and running it through the sequencer that reads through lasers, reads the excitation of each of these colors, and the computer spits out this huge sequence of A's and T's and G's and C's. That now the question is, what does it mean? What does it say? We've got all of the letters, but what are we really looking at? Where are all of the genes? What are these genes coding for, right? So this first bit we think of as genomics, and then thinking about what's up with it, that's the proteomics, right? What's it really saying? Build me this or build me that. What is it that you're actually building, all right? And you go through here and try to evaluate. Bioinformatics uses all sorts of computer programs to look at sequence data and help you to figure out, are we looking at a gene and details about it? What sorts of functions might we be seeing, all right? We talked about those amino acid tables and start codons and stop codons and all of that stuff. We're doing some surveillance as we're moving through there, trying to find these specific signatures to know. Looks like we might be a specific gene, right? So this Sanger sequencing was what we were really talking about. This is the idea, all right? So we've got our normals that could be linking up, right? And here we're showing a handful of bases. But our normal or deoxynucleotides linking up, and then the others, or maybe these are primers, whatever it is, all right? These are the ones that are the specific tags, right? And you can see as we move through here, there's some variation, right? As far as where this potential one shows up, right? Bigger bits, littler bits, as it all moves through there, as we can see this going through the detector, we piece it all together. The computer pieces it together. And in turn, we get the big sequence that's spit out. So this another story moving through here. So Javier has some intestinal issues going on. Clostridium difficile, bad news. We'll talk about some details with it in the last chunk of material, right? So here we've got some PCR products from a handful of different strains, variants of Clostridium, all Clostridium difficile, but subtle differences, ribotypes, all right? So here's Javier's sample. We've cultivated up the Clostridium, extracted DNA. Now we're looking at these patient versus a handful of culprits to really kind of clue us into what type of Clostridium we're looking at. And the specific bands from Javier are an exact match right up here to ribotype 27 match. And we know Javier has this specific strain of Clostridium. And in turn, there might be some variation with each of these things that might dictate different potentials for levels of treatment, specific antibiotics, or maybe other virulence factors that could be a potential with that. So really just sort of depends on those situations. And that's what we're seeing here with the genome and the specific fragments that wind up getting copied out of that for its respective ribotype, right? Each one subtle variation with that would show up. So here's some transgenic stuff, some eukaryotes. These fluorescent green proteins are a protein that has been, well, recognized initially, but isolated. And I think jellyfish were some of where this fluorescent protein was coming from initially. It in turn is used as a marker in all sorts of molecular biology to see, did this thing function properly? Did it get in there? If it's glowing, it worked, right? So kind of a bit spooky. I feel like this should maybe be Halloween as we're talking about this lecture, right? With the little mice glowing in the dark. Their eyes, their nose, their tails are glowing. Two glow, and the middle one, it's just a normal white mouse, all right? Didn't get transfected, all right? So getting that gene in there. Here we can see the fluorescent protein in bacteria where these colonies are glowing, right? I feel a little more uncomfortable seeing the glow in the dark mouse than seeing bacteria glowing green on the Petri dish, all right? Just a little, it's a little odd, all right? And over here, we can see a different type of media, XGAL, where we've got different colonies. And we talked some of that stuff before, where you could see based off of the colonies, different ones showing that it's inserted and it's antibiotic resistant, so it's functioning, and we can pick that off and cultivate it. I believe these ones are the ones we're looking at, right? No, blue colonies. All right, blue, white. So this is back to the lac operon that I posted those details with before. All right, so this, we saw this in the text, all of this stuff. Oh, there was this. All right, that's the end. Questions or concerns? The exam is set to open in 15 minutes. Closes Monday at 5. Have a great weekend. Peace be upon you. Farewell, YouTubers.